Yeah, you could share your screen now. Well, let's
Let me just here. No, you didn't allow me to. You have to allow me.
Okay, wait one moment.
No problem.
Yeah, it's allowed. All participants are allowed to share.
Now it is.
Oh, okay, start starting. Okay, one more. Okay, I made it multiple, so that's why it's not allowing. Okay, fine. Now it's one per.
Can you see?
Yeah, I can see. That's good.
So
I was just trying to pre-order Samsung S22 Ultra, S22 Plus Ultra.
That's good.
So
tell me.
Yeah. Today is the epic release. What they call it? Unpacked epic release. So some event is going on currently. Yeah. So Anna is part of that team, I guess. The launch team.
Oh, good.
She deals with the latest and greatest of these gadgets. Okay, let's go to our stuff. Yeah.
So the thing I added was the lab studies, this part, and I made the main topics of the things, and well, explaining the process.
Cell-based assay. Yeah, that's good.
As I wrote this here in the comments, one of the main losses are at financial time.
Yeah, like after 10 years, years, if you fail, it's a huge loss for the whole scientific community. It's a huge loss for the whole scientific community, not only for that drug organization. It's a loss for it's a time lost. Time is money.
Yeah, it's financial time and resources, because the resources are lost. And this is on the initial steps.
Very good. Your quote is very good. Fail early, fail fast. So within within a few weeks, we'll fail and we'll find out whether the drug is useful or not.
I found it like fail in some article that says that fail early, fail fast has been the mantra in the pharmaceutical industry.
Yeah, pharmaceutical.
So that's why I put it here, because.
But if you want to learn a new skill, this is not the mantra. Let's say if you want to learn skiing, if you just fail early, fail fast, you keep on falling, then you'll lose interest. So you'll have to get control, slow descent, and then you'll have the interest, then increase speed. So fail early, fail fast is only for pharmaceutical industry.
Yeah. It's a mantra for pharmaceutical industry, only that.
Yeah.
And the thing that I choose to give more importance was about the lab studies, as you told me to do, because it's the earlier steps of the whole process. So
immunotoxicity test.
The drug discovery.
Where is the cell culture? You're not written anything about cell culture.
Well, here.
Okay, here you're written cell-based assays, but okay. So after the toxicity test, then you have a cell culture. Yeah.
The thing is, when drugs fail, the two how do you say? It's mainly due to the lack of efficacy and toxic effects. So a suggestion that we can make to improve is to use cell-based assays.
Phase 3, right? Efficacy comes in phase 3.
The thing that you told me to do, it was on the earlier the earlier steps.
No, is it a closed loop approach? In a sense, you have phase 3 and the efficacy is less. Then do they give a feedback loop to the lab studies again and try and
no, not these. This is I mean, the efficacy is evaluated on a later
after
phase.
15 years.
But
okay, go ahead. Yeah, please. Yeah, you want.
But the thing is to propose to use cell-based assays since the beginning that can.
Currently they're not using cell-based assays or cell culture. They're not using. I heard
only in some drugs. Only in some cases, and the thing that I've the studies that I've found are really recent, are from 2020.
What prevents them from using cell-based assays? Is it costly?
No, that's the thing, and you can see here. They are using. They start using 3D models now more often.
Synthetic models or real models?
No, with human cell.
No.
With human cells,
I'll tell you what is synthetic model. I guess you do not understand synthetic model. Let's say if I'm training object detection using training data. If I have live video, if I have video footage from the road and vehicles, that is live data, live training data. If I use simulation, if I use a simulated environment, if I have a gazebo environment, let's say gazebo environment or any 3D, let's say through Maya, I create some environment, that is synthetic data. Through ANSYS, let's say through ANSYS, through ANSYS, if I create some world of my own, that is synthetic data. So similarly, are these models synthetic models or real models?
Synthetic models, because they construct organoids, what they call as organoids, for example. Yeah.
Organoids? Okay. Sorry. Okay.
It is like a ear or a liver or whatever. They create that in the lab.
In 3D modeling, there are many types: polygonal, mesh, some of that I just forgot. I'll show you that slide which I have for all the types. So what kind of 3D models they are using currently? Is this polygonal mesh or?
It's not like that. In the pharmaceutical.
Okay, humanoids is organoids is a completely different concept. Maybe I don't know. We'll study about organoids.
Yeah. Let me look. Here you have. Let me see where
this whole stuff, right? This whole phases and this clinical trials lab results can be modeled in terms of a controller modeling using control systems, and things could be made faster. So that's another approach. I'm trying to think about it. So you're talking about some feedback loop, right? So after 15 years, you give a feedback loop. Then the person who created the cell culture or the toxicity test would have left the lab, or you would have left that organization. So after 15 years, I don't know what are they trying to give a feedback in case in the current existing systems. So we will have to make it fast and more controller based modeling. So we'll deal with that. Okay. We'll discuss. We have time. We have time. We'll discuss all these things. I want some answers for some specific terms. Like we were discussing about efficacy, right, the other day. What is efficacy?
Yes. It's the. Let me see if I have it here. I don't know. I don't think so.
So can we do
It's
rapid prototyping?
it's measured with the success that it's
Is it an index? Is it an index? Okay, yeah, you explain.
How can I explain you? I'm thinking the way.
Okay, make it very simple in the sense.
Yeah.
There are.
It's
Okay.
so
It's
when you
probability.
design, yeah,
It's a probability. It's a probability.
it's not a probability. It's like yeah. After all, it is a probability, because
So let's say 89%. If you say 89%, out of 100 times, 89 times you will survive. 11 times you will get the disease.
Yes, it's like when you design the clinical trial, you define your efficacy like according to the target, according to what you have designed. So if
okay,
you
fine.
design,
It's okay. If you understood efficacy concept, that's fine. You need not explain to me. I just want you to understand so that you will make things faster. So you will not explain to me. I'm not going to explain to Lubrizol.
Okay.
You will be there. So you explain to him in a way you can understand.
Okay.
But always you should be able to explain the concept in a way that a 5-year-old can understand.
Yeah, that's why I was
only then you have understood the concept properly. Otherwise, you have not understood the concept properly.
Yeah.
Let's assume that I am a 5-year-old. Okay, we'll go to efficacy later. But what I was trying to wait, I just missed out that topic. Yeah, can we do rapid prototyping? Yeah, that's what I was thinking about. Can we do rapid prototyping? In a sense, this is the complete behavioral model of the drug discovery. Can we do a rapid prototyping, which before we actually deploy, in a sense, implement this whole pipeline?
From what I've seen, there are already some prototypes commercially available. So some of them are easier to create than others, mainly because to test a drug, in the case, you have to consider the metabolism, so you have to involve medicine, because you have to know what's the aim of the drug. You have to know the metabolism of the drug in a certain organ or whatever type of cells. So since there are available, there are commercially available.
What software?
For example, kidneys, there are what they called something like tumoroids or something that are for tumor cells, cell lines.
Tumoroid, that's what it is.
Tumoroid, something like that.
Okay, you message me later.
With tumor, well, I don't know. I don't know how it's
message me
spelled.
Later. So no, I want the software so that I could try out. I'll just get some evaluation versions and I'll try out some rapid prototyping.
Okay.
So if
it depends. That depends on the drug that you want.
Yeah, I'll take some simple drug. I'll just take.
No. I'm just saying that it depends on the drug that you want to test. For example, if it's a liver, I know that livers are already being prototyped. But for example, the brain, it's not. No,
so imagine we are creating a drug. Imagine we are creating a drug. I want to do rapid prototyping of it.
I have here, I think. They have a platform to facilitate the decisions. I have one of the sites. I can message you later the link for it. Actually, one of the, let me see, I think it's this one.
See, if they are already doing rapid prototyping, so these organizations already know the concept of simulation. They're just not doing it for some unknown reason.
Yeah, that's the thing.
If they're already doing rapid prototyping, then what prevents them?
I think that they are being used for research purpose.
Feasibility
not
test.
for sorry?
Feasibility test.
It's like academic researches, not
scale, industry scale.
Yeah.
See, we are an organization. We are a hybrid organization in the sense we work like academic institutes as well as.
Yeah, but the thing is to connect both fields, both academic and pharmaceutical, in this case, fields, because.
Yeah, academics is not greedy. Yeah, pharmaceuticals are greedy. Yeah. So we will avoid all the greediness from this pharmaceutical industry.
That's
so
the thing.
We would collaborate with academics and do all the research.
So here you have commercially available solutions for 3D. You were asking me what kind of
software
structures, models that they were.
Just show me share your screen. I guess you have shared only one.
Okay, sorry.
The entire screen. Show me the entire.
Yeah. Well,
unless you want to hide that software.
No. Okay. I thought I was sharing it with you. Sorry.
That's fine. We will buy all the software. If you have some proprietary software which you want, I'll fund that. No, don't worry.
Now I cannot. I'm not being able to.
Yeah, and one more advantage is you're kind of a student, right?
More or less.
More or less. So you get all the advantages in the world in terms of software. So wherever you go, if you just register as a student, you'll get.
The thing is, now I'm not affiliated to any university, so
you'll have your student email ID, right?
Sorry?
You'll have your student email ID, right?
I do. And I still use it.
Judicious manner. Okay, I should not be telling this.
I don't know why I'm not being able to. Oh my God. I don't know why I cannot.
Okay, just stop sharing this screen and share that screen.
No, because I have sorry. No. It was something stupid because I have a monitor.
It's a monitor is a unnecessary unnecessary distraction. I never use monitors. It's an unnecessary distraction.
Because it was easier for me to work with the monitor, and now I wasn't finding the.
If you start looking at the monitor for a long time, then you see something else. Everything look miniature. So this is the same effect.
That's the thing. Yeah, but I was not looking for the monitor for the sharing things.
Yeah, I'll tell you the analogy. Like if you watch a movie in the theater for a long time and then come out, all the people will look as miniatures. So it's a similar analogy. So I always prefer laptops and.
So I thought I was sharing this with you. Now you can look. Now you can see.
No.
No, really? Oh my God.
Yeah.
Now you can see.
Yeah.
Here the commercial available solutions. There are several type of 3D cell cultures, but these ones are the most. It's an enterprise.
Okay, tell me the software's name of the software. I'll just search it and install it in my system.
Okay, they have several here several things that.
Or just share me this document.
Yeah, I will send it to you. Yeah. But you were asking about the model, the most common models they are using now. It's this one, 3D cell culture scaffolds.
Okay, what the cells they have?
Sorry?
What cells they have in this scaffold?
Human, I'm not, I think is this one, let me see.
Primarily, I want heart and brain cells. We will deal with the rest later. Or even if you have stem cells, it's fine. Adult or embryonic, anything's fine. Organoid techniques, very interesting. Stem cell and organoid. Oh, okay, organoid is something new for me. I have to learn about it.
Well, it says in one of the articles I have.
So now this opens a new avenue. Like organoid is like the equivalent of a robot in the macro space to a robot of a stem cell in the micro space.
Yeah, more or less like that.
If I'm not wrong, more or less, if I'm not wrong. So organoid is going to be the humanoid of stem cells. So stem cells is intelligent like humans. So organoid is going to be super AI. So it is going to be more intelligent. So we'll deal with organoid. So this will help us in terms of creating nanorobots. So organoid can be nanorobots. Let's discuss about that.
Yeah, one of the things that you've told me was about the nanorobotics. Yeah. I have that. I looked for it and they are already trying to use.
It's all in theory. I've already researched it. It's all in theory. It's none of it is in industry scale. So we are the first industry to bring it to industry scale. We are the first organization to bring it to industry scale. So I have a meeting with the Pittsburgh Robotic Association next week, and with probably some guys from Carnegie Mellon will also join that. So I will discuss all these topics.
Okay.
I will collaborate with them and we'll bring nanorobotics to industry scale. I'll also discuss about organoid. Okay, before that, I have to do some research about organoid. I know about stem cells. I don't know about organoid.
Well, the scaffolds can be from biological origin.
Organoids are miniature organs. That's pretty interesting because stem cells are not miniature organs. They are the blueprint of organs. Stem cells are blueprint, but organoids are miniature organs. So this is pretty interesting in the sense we could mimic the behavior of the organ. We could just take it out. We could just take out the organoid and mimic the activities of the organ in object. An object can be anything. It can be even a humanoid. So a humanoid with all organoids. Imagine that. So that would be a real human.
Yeah.
Okay, that's a different topic now. I'm digressing. So now let's discuss about. Okay, now we have the software. We have the rapid prototyping. I'll do some rapid prototyping. I'll understand how they are doing it and how they are doing the feasibility test and why it is taking so long. If they have already have software for rapid prototyping, then modeling would be very simple. They have the models, everything with them. Then everything could be model-based development. There's no point in doing all clinical research with human guinea pigs. So I'm sorry, I should not use the term. We should do everything in model-based development, and after that, use that parametric model, its behavior as input to AI model, which gives us the final result in terms of either the probability. We look for only efficacy, right? What else is the output of phase 3? Efficacy and safety. Okay, safety percentage. That's again a percentage, right? Safety.
Yeah.
So safety and efficacy. Those are the only two outputs of phase 3, right?
Yes.
Okay, then I'll create
basically
a model.
because you measure the phase tree with the actually in the phase tree you have the disclosure of less common effects, but the evaluation, the only evaluation that you have,
Okay, what does FDA
efficacy
need?
and
Okay.
safety.
Oh, I'm not worried about all the parameters. We just make it very simple. What does FDA need? What parameters FDA need to approve a drug?
This slide is based on the FDA
guideline
rules. Yeah.
Okay, forgot the term. Okay, fine. Okay, so FDA just needs efficacy and safety. We'll just it's a very pretty simple AI model. It's going to be very pretty simple, and even this model based development controller architecture would be very simple. I just need some models, either a humanoid or stem cell. We'll create and some rapid prototyping, so I'll understand the software. So do you have software for model based development as well apart from rapid prototyping? For the controller modeling, rather, I used the wrong term. For controller modeling, do you have any more software?
Sorry?
For controller modeling, do you have any software?
No.
So they don't do any model-based development at all?
Not that I know. I don't know. I didn't search for it.
Okay, we'll come again to that later. This is quite good enough to initiate our discussions with Lubrizol. And I'm feeling more confident. I'm feeling more confident now that we could have a real solution. Till now, till the last meeting, it was slightly utopia, but now we are closer to a real solution. So we'll have a real solution soon for controller modeling of the drug discovery pipeline. And since we have all the models, so it's a good work. You have all the models, all the information about the models. So you have both animal models and okay, what's the advantage of animal models versus the cell-based assays? It's not close, right? Animal models is not close to a human.
The thing is, using cell-based assays, it involves actually it can involve both human primary cells and 3D models. Even the 3D models can be synthetic or with human primary cells.
What do you mean by primary cells?
It's of human nature. So probably they collect. I didn't search for it, but they collect human cells and they culture them.
No,
but
no.
I will look for it.
Why is it called primary? Is there a secondary, tertiary?
Oh. Let me see if I.
Okay, how long does a culture take place? What's the duration of a cell culture? Normal. I don't want to fasten it initially. Let's keep it normal.
Let me just.
This is going to be pretty interesting. See, 3D models which are going to.
That's what I was saying. Primary cultures are cells freshly isolated from organ tissue and maintained for grow in vitro.
And what's secondary and tertiary?
Huh?
What is secondary and tertiary then?
I didn't understand. Sorry.
Okay, primary is like you're taking it fresh from organs, from the things. Yeah. What is then secondary, tertiary then?
Probably it's a subculture from the first one.
Okay, fine. Oh, you're reusing the same cells.
It's probably. Let me see.
Okay, yeah, you can find out later. So 3D models mimicking human physiology. Can we bridge holo anatomy to this? So holo anatomy is going to do something similar. So there again, I guess it's a synthetic model. I don't know. Like I have to check holo anatomy. I've not checked it personally. But it's going to be kind of a 3D model, a hologram model instead of a 3D model. It's going to be a hologram model. A hologram model mimicking human physiology. So you could add that as well to your slide. So we'll make it more elaborate, just to show that we are also using Microsoft technologies for our drug discovery. Okay, that's later. We'll add it before the Lubrizol meeting. So primary cells. Okay, tell me, what is the duration of a cell culture?
It depends on
the type of cell.
Yeah.
Okay, let's take heart cell. Okay, does
9 for 14 days.
14 days, yeah.
Of ventricle.
Fortnight, yeah, I heard this from my friend. He told it's a fortnight.
From the ventricle, for example.
A ventricle. Okay, so a heart is a very complex organ.
Yeah.
How many cells, how many blueprints make a heart? You understand my question, right? How many blueprints make a heart?
I don't know.
How many stem cells make a heart? Okay, I was dealing with these things. Perhaps I will have to check again. So similar to neural networks. A stem cell can be programmed to be any organ.
Yes.
Yeah, a skin stem cell can act as a heart. Rather grow as a heart. So the advantage of this is, imagine we don't have stem cells of a particular kind. We just have skin stem cells. We just have skin cells, stem cells. So we could just use skin stem cells and create all of the organs. Do you agree with that, or it's wrong what I'm telling?
I don't think it's
possible.
No, I don't think it's wrong. But I'm not sure if it's that easy to do that thing that what you were saying.
Easy and hard. Impossible is a term.
No, not impossible. I don't think so.
If impossible is not blocking us, if it's not impossible, okay, even we work on concepts which are impossible. So for us, hard is not difficult.
As you told me the last time, we are looking for the future. So no, yeah, it's not impossible. It's not the word.
Okay, Walt Disney, you might have seen the quote of Walt Disney. Doing impossible is fun.
Yeah, it is.
So we'll do the impossible. So we will use one type of stem cell, whichever is available easily. So a skin is very available pretty easily. We could just get skin cells and develop all cultures. The reason why I'm talking about that is, if you want to do on a large scale and if you want to fasten the process.
I think that's one of the most hard things to do until now. It's to enlarge the scale.
Yeah, we'll do an industry scale. We have all the funding. We could justify this funding. Okay, I need all the complex topics in the world. You know why?
Why?
Only then I'll get funding for $6 billion. Yeah. If I do some simple stuff, then they'll say, oh, you're just doing the simple stuff. Why do you need so much fun?
Let me show you these. I think it's this one.
I have to justify my funding. So I will have to do the most impossible stuff.
Let me see if it's this one. Yeah. Here, let me look for it.
There is a 2D and 3D cell culture as well.
Here. Utilization of stem cells fabricate 3D spheroids and
what is spheroids? Organoids, I understand. What is spheroids?
Spheroids, I saw that. Let me see. I have it somewhere. Nope, it's not here.
Cytokines, robusty. Okay, this is a very good. This is very good. I understood the concept. So spheroids are going to be part replacements of organs. So organoids are going to replace the complete organ, and spheroids can be used for injuries. Let's say your kidney got injured, so you could replace a part of it. So this is very essential when you don't want to do surgery and you just want to do a non-intrusive way of curing organs. Do you agree?
Sorry, I was looking for the thing. Tell me again. Sorry.
That's fine. Spheroids can be used for repair of organs without replacing the whole organ.
Yeah. Because it's an agglomerate.
It's like patch.
Let's
like
say this way. Yeah, it is.
So this is very good. So
it's in the cell aggregate.
So there are three kinds of cultures, as I understand: stem cell culture, spheroid culture, and organoid culture.
But the thing is, with the organoids can that's why I was sharing this with you, because they spheroids and organoids can be used, can be fabricated, can be done with stem cells.
Okay, so we just need stem cells.
They use the stem cells to do the organoids and the spheroids.
How difficult is to create spheroids and organoids from stem cells?
That thing, I don't know, but let me see.
The more difficult, it's more interesting. If you tell me it's difficult, then I'll be interested in it.
Actually, I'm not sure if they are already using the for the 3D. Oh yeah, they have. Because 3D, of course, it's completely different.
Awesome. Organoids can be used for treating Parkinson's, Alzheimer's, autism. This is pretty awesome. Where did you find this article?
Well, I did research for a long time, and I had to
write
my PhD thesis. So
yeah, fine. Yeah, you have done the thesis on this. Okay, fine. So
fortunately, I have a great how do you say, a great way to search for the information at academic level.
You search the scholarly articles. Okay, I'm a pretty basic guy.
It's not the scholarly.
Yeah, I just search the general Google. I don't search scholarly articles.
Yeah. I did the research as I was doing for my PhD. So
okay, fine.
Because I had to support everything that I was saying. So
that's good. I guess you also have a Google Scholar account.
Yeah, I have.
Yeah. So let's concentrate on this. Is very essential for me now for the current project.
I can send you the articles that I found.
You have. Okay, just put me on a Google Drive and send it across. So I need organoids, especially because I'm going to adopt a child. I'm going to adopt an autistic child.
You are?
Yeah, I'm going to adopt. Yeah, I'm going to adopt an autistic child. It'll happen within a month. Not necessarily high functioning. He need not be high functioning. He may not do the day-to-day activities, but still I'm going to bring him to the mainstream schools by way of technology. So this organoids and AI is going to help the autistic child to behave as a normal human being. So
yeah,
so this organoid is
that seems interesting.
That's pretty interesting. We'll bring all the people with different who are differently abled. Don't call them special. Who are differently able to the mainstream.
So in some years from now, you'll be able to, or we will be able to treat my joint problems. I'm being followed on the rheumatology since I was 27, and they didn't got me diagnosed yet, and it's being really
come on, we are talking about heart ventricle stabilization and non-intrusive heart transplant. We are talking in those terms, and you are just talking about some silly stuff like joint pain.
No,
so
I was saying because it's an autoimmune disease.
No. Though
And
anything can be cured.
so,
See, we are talking about nano robotics, which is going to
I know.
do the cell dynamics and going to cure a lot of stuff, do the repair work.
I
This is
know.
all silly stuff. Joint pains is all silly stuff. We are talking about something serious like Alzheimer and Parkinson disease. So I'm actually a part of an organization which does Parkinson's disease research. It's called Friends of Parkinson Disease. So I'm the board member. I'm the board director for it. In the sense, I give advice to them. So I can give a lot of advice to that organization based on this article alone. I'll give the credit to you. Don't worry.
No.
Okay.
I was
So
just
Alzheimer's,
reading.
Parkinson. So this organoid is going to organoid is so organoid is a revelation for me now. Today I learned something new. Organoid and spheroid.
I was just reading the paper. Here.
Okay, pluripotent stem cells. Is it difficult to create or to get? What is it? Pluripotent stem cells.
I don't think so, because
I know adult stem cells, embryonic stem cells. I don't know what is pluripotent stem cells.
Let me see.
Are these things all real, or just for getting thesis through? They just write some stuff.
No.
It's all real, right? So they should be validated with proper experiments or stuff like that, right?
They have to. The thing is now it's more difficult to publish anything, and they really do
a lot of validation. Reviews. Yeah, I understand, because I submitted an article for SAE and they had some 10 reviewers. They had 10 reviewers with various industry background and expertise reviewing my paper. So yeah, review is quite good.
A colleague of mine tried to publish an article, I think, for 4 times now, and she couldn't. She wasn't able to because of the statistics and everything, and
no,
I completely agree.
Sometimes the reviewers have a lot of ego, and they'll try to show it through rejections. So that doesn't matter. For us, we need raw ideas. You understand what is raw ideas, right? So raw ideas comes
of course.
Raw ideas come.
Because you have to innovate. You have to present something new.
We will fine tune it and polish it. We need raw ideas from kids. Kids have raw ideas, so we need that kind of mentality. So we should develop those raw ideas into achievable results. So pluripotent, just research on pluripotent stem cells, because we want to create a lot of organoids, and we are going to cure a lot of diseases through organoids and spheroids.
So pluripotent stem cells are so-called master cells. They are able to make cells from all 3 basic body layers. So they can potentially produce any cell or tissue the body needs to repair itself.
From where they're taking? From where they're taking the pluripotent stem cells?
Let me see.
And is it available for adult stem cells, or it is available only for embryonic stem cells? That is also one question. See, just this through this one article, I'm able to solve all the diseases in the world. I don't understand what the guys of the scientific community doing when they have all these articles.
Sometimes it depends on the budget that you have.
It's all budget. See, I don't worry about it. It's all budget. It's all greed that's preventing our society from curing all diseases.
At least in Portugal, the academic field, on the research field, it's really hard to develop because of the lack of
see
of money,
and
of
one more
budget.
thing, robotics need not be physical. You understand, right? Robotics need not be physics.
Yes, yes.
Robotics can be biology as well. So we can bring the concept of biology for robotics. So we are going to place organoids and spheroids as nanorobots in the bloodstream. So that's my idea. One moment.
No problem.
Okay, so yeah, I will propose this idea to Pittsburgh Robotic Association. We are going to place spheroids and organoids in the bloodstream to start with. And are these entities intelligent? Do they have intelligence? In the sense, do they act on their own?
I don't think so. I think they are.
No. The reason why I'm asking, a cell can act on its own. In the sense, a cell has intelligence. Does these organoids and spheroids act on their own?
That was I was saying. I don't think so.
And how are they preparing stuff? How are they growing? So if they are growing, if they are growing into an organ, if an organoid is growing into an organ,
it's should not have some growing to an organ. They are placed to. They are constructed. How can I say? At
it's
least
like a graft. Is it a graft? Like in the sense, your bone is broken, and you just tie the bone in such a way that it naturally grows. Is it something like that? Is organoid something like bone healing?
I think so. I didn't see that.
So bone healing has no intelligence.
Yeah.
See, okay, bone healing has no intelligence. So organoid does not have intelligence. That means. Okay, do some research. I've given a lot of tasks for you. So find out whether organoids can be used as a single entity like cells, and whether they have intelligence. So now we could introduce artificial intelligence into the organoids and make it a physical robot instead of from a biological robot. We'll think about that.
You were asking me about the pluripotent stem cells.
I asked you three things.
They can.
What are pluripotent stem cells? What are organoids?
They can be generated directly from a somatic cell. So, not only from stem cells.
Oh, that's great.
It can be generated by I don't know how to pronounce it. Gametes.
Spell it.
Gamete.
Spell it. Put it in the chat window.
Let me just. Oh. So it can be. It can be from this.
Yeah, I know this. Yeah. Undifferentiated stem cells. Okay, which is the largest source of these gametes in the body, in an adult body, not in embryonic, in an adult body? Which is the largest source of gametes germ cells, which can be easily separated. I'm asking you, which is the largest
so
source?
no, you can only the undifferentiated stem cells in even all the other can be retrieved from an adult body.
Which part I'm asking? Just the skin?
No. Actually, the first one that I don't know how to pronounce, it's
Forget about pronunciation.
From the sperm on the males. Let me see if and the oocytes from the woman.
Okay, the womb is difficult. Sperm is very simple. So just from the sperm you could get all the gametes.
Yeah, because the gamete is it's the how do you say,
the blueprint, the undifferentiated stem cell. Okay, I understand. It's natural because sperm is the life producing liquid, and it will have the undifferentiated stem cell. It's pretty natural. And womb again, it's pretty natural, but getting it from womb, it's difficult. So how do you in an undifferentiated okay, then the sexes also would be it will be unisex, right? These gametes would be unisex. Is that?
I don't know how.
Does it even
I was.
Okay, yeah.
No, I was trying because I don't know how they can produce pluripotent stem cells from those.
Okay, that is I'll research on that, but this is a good starting point. Like, does it even matter that the stem cells be unisex, in the sense organs does not differentiate between sex, right? So the organ could be same in the sense of a male can do an organ transplant for a female. I've seen a mother donating kidney to her son. So it's this doesn't matter whether we take the gametes from a sperm or from womb. So we can easily take it from sperm. So this can use for all pluripotent stem cell research and organoids and spheroids. Is that very simple, or I've oversimplified stuff?
I think you are just simplifying stuff.
I want it simple. Like, simple is always effective. Simple is always effective. Yeah. You know why people make it complex? You know why people make it complex?
Sorry?
You know why people make it complex?
Because they don't think simple.
No. Because they want to appear as if they are very intelligent.
Yeah. It is true.
Yeah, let's say all the PhD guys, I don't blame you guys. The PhD guys try to make it very complex just to show that they are very intelligent. But everything's very simple. I can tell you that. Everything is very simple. Even this pluripotent stem cells and all the organoids, the spheroids, we can do it in a simple manner. So this is a good starting point. So thanks for giving all this information. My friend was not discussing about gametes anytime. He was always discussing about adult stem cells and embryonic stem cells, and from there he started. So I was missing some part of the story. So now I've got
but I also learned this today still because I didn't know.
It's on the job training. Yeah, that's good. Okay, we have learned many things. I guess we can solve all the diseases in the world. I'm not kidding. We can solve all the diseases in the world just by a gamete.
I was trying to look to see if the process
is that simple. Okay.
No. To induce the pluripotence of the stem cells.
It's very simple. Just contact a sperm bank. There are many sperm banks available. They just use for IVI, in vitro fertilization. But they don't use for other research. Now we are going to use the sperms for Pluripotent stem cell research and organoids and spheroids. It's pretty simple. Do you agree? Or I'm just making oversimplifying stuff? Even
no,
if I oversimplify,
you are making me
because,
believe that it's really achievable.
Yeah, that should be the case. I'll tell you. If you oversimplify stuff, the advantage of that is you will be very confident and you will start doing things. Let it get complex as and when you go. That is fine. But still, we are starting it. So well begun is half done. So well begun is half done. So we will make things very simple and start doing it. So just contact a few. What you will do? Okay, you don't have any lab facility, right? Are you associated with your university till? Will they allow access to their labs?
I'm not affiliated anymore.
Okay, fine.
But if we have a concrete design,
collaboration.
Okay, just no. I mean a design, a test and assay, all designed with the steps and everything. I can present the idea to some
or dean
universities.
Don't go to some university. What about your university?
No, the thing is, my university, it depends on the issue, on the groups. They have a small group on the health department, and I'm not sure if they work with those kind of things. But I can go to the University of Porto. It's 100 kilometers from here, so it's okay. Or to the
I was doing this activity, like
University
I traveled.
Of Coimbra, so.
Yeah, I traveled all over Germany. When I was in Germany, I traveled all over Germany to find more about epigenetics, just for the sake of my friend. My friend wanted to join a university in Germany, so he wanted to know more about the university. So I went around to all the universities and I inquired what they are doing. So that's how I knew about epigenetics. Otherwise, I don't know anything about epigenetics. So everyone is so interested in explaining to me, even they knew that I am coming from engineering background. I knew nothing about zoology or epigenetics. So they were explaining to me in great detail. So I went to Göttingen. I went to Max, Stuttgart University. I went to Tübingen. I went to many universities. So I know everything what's happening in Germany. Maybe I'm outdated now, but I know what they were doing then. So you find out what they're doing in Portugal, or if you have the opportunity, you are a EU citizen, right? You can go to ETH also. I'll sponsor the trip. Go to ETH Zurich. I love ETH Zurich because they do some extraordinary stuff I knew from my friend. Go to ETH Zurich.
Can you write the name in the chat, please?
Oh, come on, you don't know ETH Zurich and you call yourself a microbiologist?
I'm not sure if I think, yeah.
This is a premier institute. You should be knowing about it. Okay, so go to this place. If you want a video conference, I'll do it from here. We'll discuss all these things with them and we'll get all the access to their labs. The reason why I'm asking you to go here is you'll have the best labs. We can get access to all labs and do all the experiments with organoids and spheroids and pluripotent stem cells.
I think we have to have a really well-designed experiment before we go to any research.
Like, what is that? See, we are not doing the actual complete. We are going to explain the idea. We can ask someone from their lab.
I know, but you have to have.
One moment. Listen. I hate when people put down the idea. Just throw away the idea. I hate it.
I'm not putting down the idea. I'm just saying you that
listen. Listen. we will have someone from their lab. We will have someone from their lab doing the actual stuff. We will just give them the prototype and the guideline. It's like in your Google navigation, you changed the text version, not the GUI version. You change the text version. It will say after 2 kilometers go right, after 3 kilometers go left, something like that it will tell. So similarly, we'll just give the directions to the lab assistant, whoever is doing that experiment. He will have more information on how exactly to do that. So we will provide the storyboard, and he will do the actual experiment. So the storyboard is simple. Extract gamete from sperms, extract pluripotent stem cells from gamete, and create organoid and spheroids from pluripotent stem cells, and use the organoid and spheroids. Okay, we'll start with spheroid, simple. We'll use the spheroid to do a repair work on an existing organ. This is the storyboard. And anyone who's in the lab can understand what we mean by this. So they're going to do this. And we'll just take some simple organ, a very simple organ, and we'll try to repair it. It will take less than a fortnight to prove our results. Just go to ETH Zurich with this idea. I'll sponsor your trip. I guess for you it will be pretty simple. Switzerland is just nearby, right? You can go by.
I think it's maybe 2
hours, intercity.
2 hours.
Yeah, intercity express, the IC, if they have it in Portugal, the fastest train. Or the
no, they don't have.
Yeah, which is available in. No, it's not the fastest train. The fastest train is with France. It runs 500 miles per hour. The IC runs at around 300 odd miles, 320.
No, but we have how do you say low cost.
I know. Yeah. See, you'll get a Eurorail pass. So if you get a Eurorail pass, you can go anywhere in Europe with that pass and have fun.
That's true.
So the reason why I'm asking about train is you'll enjoy the ride. You will see all the scenic beauty and stuff. If you go by flight, you do nothing.
Yeah, but the travel is longer.
No, travel is not longer. I bet. See, you go to the airport, you wait for 2 hours, you check in, and you then fly again another 2 hours. Then again you get down, you unpack all the stuff, another 2 hours. So 4 to 6 hours. The same 6 hours, in less than 6 hours, you can travel by train.
I don't think it's enough to
just try out. Just try out. For Zurich, it will work. Just try out the time difference between train travel and flight travel. It will be you'll find out which is faster. Okay. I guess this is enough for today. You just send me these articles. We have done a lot of groundwork. We have done a lot of work. I'm very confident that our utopia is going to be reality pretty soon, and I'm very confident that we can solve all the diseases in the world just by these 3 entities: pluripotent stem cells, organoids, spheroids, and that's going to assist nanorobotics as well. This is pretty awesome. I never thought biology can be so interesting. In the sense, microbiology can be so interesting. In the sense, my friend did not give me the complete story. So you're giving the complete story. It's very interesting. Yeah.
Well, my thing is due to my PhD, to my work in research field. I discovered that programming is not that bad also. So.
Yeah. So everything is programming. Even your genetic code. Even your genetic code is kind of programming. So the reason why we were able to do a genetic map of the whole human body at a faster pace was due to the crowdsourced structure. It was crowdsourced, right? The genetic map of the human body.
It's what?
Crowdsourced. You understand the term crowdsourced?
No.
You understand Linux?
I know Linux, but I never
So
worked that much with
Linux became so famous because it was crowdsourced.
Yeah.
So similarly,
Yeah.
genetic map of the body, or the human body.
Yeah, you told me last time.
It was crowdsourced, I guess, if I'm not wrong. Genetic map was crowdsourced. So that's why they were able to do it at a shorter time. Shorter time is like 20 years. Okay.
Yeah.
Okay. When was it released? I forgot the date. The
Sorry?
year. Genetic map of the human body. The complete genetic code.
I don't remember.
Yeah. A few years back. Yeah. So we need that as well. If you have that database, give me that as well.
I will send you one of the articles that I show you. It has the commercially available solutions. It's this one you want?
Put it in chat window. I don't know where you're putting it. Okay, I have.
Is this one? No.
I have another. Okay, no, that's fine. You continue. That's not that important. Those guys are just wasting my time. See, everyone, every supplier just comes and would you like to know more about us? Would you like to know more about us? And they won't have any content at all.
Let me see. Maybe it's this one.
Commercially available what? Software. Are you shy to share these articles or documents?
No. I will send them to you. I'm just trying to find which one do you want, because
I want everything. If you have a whole bundle,
want everything.
If you have the whole, if you have a whole PhD thesis in one bundle, just put it in Google Drive.
No, but yesterday I selected.
You don't want to share it? Okay, you have done so much. You have spent 10 years of your research, and you don't want to share it with some person.
No, actually, it's available for everyone. So.
Yeah, then share it.
Everyone can
look
to my PhD thesis.
So that's how we save time. See, you have a lot of knowledge on some topic. You just share your knowledge. So that's how we save time. So I did not do the same thing. I did not reinvent the wheel. So similarly, I should encourage all our employees to share what they know, so that we could save everyone's time.
Is this one that you wanted?
I want everything you have.
Okay, I will send them. I will send every
I'll find out what I want. Just send everything you have. Yeah.
Okay. Yeah. Because yesterday I had to select from all the things that I've found those ones.
It'll be a good brush up for you to know what
those ones.
you're doing. Yeah.
That's why today
it'll also be rewarding to know that after some time, if you see all what you have done, it will be very rewarding for you to know that, okay, you have done so much.
No, today I've learned so much just talking with you about these things.
That's the advantage of brainstorming. See, I don't know anything about your topic.
Definitely.
I don't know anything about your topic, but still I was able to speak so much because I want solutions. So if I'm interested in solutions, I would try to find the route for it. So that's brainstorming, and we will involve many people in brainstorming, and we will involve experts like you. So we will get the solutions. So, okay, just send me everything you have, and I guess it's time for you to prepare your dinner or something. So yeah,
almost there.
Yeah. Okay, let's meet some other time when you find more information. We will try to schedule the call with Lubrizol either this weekend or next week. We'll take it slow. It's okay. We'll make it very elaborate presentation, and now I'm very confident.
Yeah, that's the thing. Maybe next year.
May not know. Lubrizol may not know anything of what we are talking. I'm pretty sure. So we are going to.
That's why we have to have all the answers.
So we will be in a better position. We will be in the driver's seat. I want that. I want driver's seat whenever we discuss with businesses, because we should give them directions. They should not give us directions.
Yeah, of course.
Yeah.
So probably it's better to meet with them next week. Yeah. That way we can still talk with each other
Okay, anyway,
about
that's
the
fine.
things that we have.
Even if we meet next week, it's fine. I'll prepare my executive summary. So the executive summary is something which the funding organization needs to know what and all projects we are working. So this is very elaborate project. So I'll prepare an executive summary based on this, and this would easily get us fetch us a few billions. Yeah, so pretty interesting. Okay, I'll allow you to continue with your work, and let's meet some other time. Yeah.
Okay.
Bye.
Bye.